everybody, and welcome to today's webinar. My name is Melissa, and I'm a Scientific Training Officer in the training team at EMBL-EBI, and I will also be your host for today. I am now going to hand over to Anya Fulgrava and Nancy George, who are going to talk to you about Expression Atlas.
Okay, I think we're not muted anymore. Great. So good afternoon, and thank you for attending today's webinar on Expression Atlas, a knowledge base developed here at the EBI which can be used to discover gene expression across different species and biological conditions. My name is Nancy, and with my colleague Anya, we are both curators within the gene expression group here at the EBI. If you do have any further questions or feedback after the seminar, please feel free to also get in touch via the email on the screen, gene-expression@ebi.ac.uk. So, what is Expression Atlas? It is an open science resource that selects, curates, reanalyzes, and finally displays gene and protein expression data. The website where Expression Atlas is hosted can be found at www.ebi.ac.uk/GXA. Our mission is to provide the scientific community with freely available information on the abundance and localization of RNA and proteins across different species and biological conditions. This allows researchers to ask questions such as, where is my favorite gene expressed? And how does its expression change across different biological conditions such as disease? So with Expression Atlas, we can take datasets from different genomic databases such as ArrayExpress, Geo, and ENA, as well as from within the scientific community. Using the infrastructure here at the EBI, we can reanalyze all of these datasets using the same analysis pipeline. We then present these results back to the scientific community to help answer their questions. So what do we mean by big data? Literally, this can mean very large data sets. This experiment is taken from GTEx, which is a data consortium that investigated genetic effects on gene expression across human tissues, highlighted by Nature in 2017. This dataset covered 550 donors and 53 different tissues. This generated almost 20,000 paired RNA sequencing libraries. If you tried to run the analysis on your local computer, it would take about 22 years and up to 78 terabytes of storage. Of course, GTEx is not the only example. We have a number of similar large-scale datasets across different species, developmental stages, and disease states. For example, in addition to basic research datasets, we have cancer research, human development, model organism development, and cell line model data. As of the latest release, Expression Atlas contained over 3,500 datasets, of which 173 are what we call baseline, constitutive normal expression datasets, and just under 3,400 differential datasets. These are datasets which explore different biological conditions, such as gene knockouts or disease states. So in this webinar, we will guide you through what these datasets mean in more detail. The datasets we have in Expression Atlas cover across 60 different species and counting, under multiple different biological conditions, with the sole aim to help scientists find answers to their biological questions. How do we do this? Gene expression data is firstly curated to produce high-quality, richly annotated datasets. These are all sent through the same reanalysis pipeline and presented in a user-friendly interface to allow data search and data visualization. So this is the Expression Atlas homepage, where you can search for your gene of interest or various biological conditions. You can also search for datasets from a specific species. You can get a quick overview of all the experiments currently hosted in Expression Atlas via the Browse Experiments tab. Those experiments with the blue arrow are what we call baseline experiments. That's expression under normal, unperturbed conditions, whereas those with the red and blue arrows, which representing over or under expression, are what we call differential expression datasets, where you're comparing changes in gene expression across different experimental groups. So, as I said, you can search for the data in a number of different ways. For instance, you can use Gene Expression Atlas to answer questions such as, where is my gene of interest expressed? You can do this via the Expression Atlas homepage. So on the Expression Atlas homepage is a search bar, and I'm using this here to search for the gene reg1b as an example. If you don't specify species, such a search will retrieve all of the data from all species currently available in Expression Atlas. So searching without specifying a species retrieves data for across all species for that gene. This is a search results page resulting from the query in the previous slide. You can see the cross-species datasets available as part of the search filters on the search results. So I'm just trying to highlight the box here. Search results are ordered by the number of datasets within a species by number of datasets. So in this case, there are more human datasets, so that's visualized first. To see other summaries for different species, scroll down through the search results. For example, if you scroll further down, you can see baseline results for monkey species, I think, macaca mulatta. Okay. So let's go back briefly to the Expression Atlas homepage and search for reg1b again. However, this time using the species search drop-down to specify that I want results only for human datasets. Restricting a gene to a specific species, such as Homo sapiens or Mus musculus, from this drop-down list will restrict your search to the ensemble gene ID for that species. So once again, this is a search results page. Notice two main differences here. Firstly, that reg1b is now restricted to the ensemble gene ID for human, and that another tab has appeared. This is the Gene Information tab. Clicking on this tab will give you an overview of the information about that gene, such as synonyms, orthologs, and associated gene ontology terms. Going back to the main search page, as I mentioned earlier, there are two types of datasets in Expression Atlas, baseline and differential. And for the next few slides, we are going to focus on the information provided for baseline datasets. Just to remind you once again, these are datasets which investigate constitutive, non-perturbed gene expression in a species. The main thing to notice in the search page is the heat map. This displays the gene expression across all investigated tissues from different experiments integrated and visualized in a single heat map. So across the top, the columns are the different tissues, and as separate rows are the different experiments. Just to highlight, T stands for transcriptomics experiments and P for proteomics experiments. Each point in the heat map reflects a binned level of gene expression. This is high, medium, low, or below cutoff. You can filter your results using the box above, which allows you to, for instance, show only those datasets where your gene of interest is expressed highly. Alternatively, you can also filter your results using the Filter Your Results sidebar, where the species was before. And using this, you can filter your using the metadata within each dataset. For example, organism part, different cell lines, disease states, or individuals. So to investigate the data more closely within a specific dataset, you simply need to click on that dataset of interest. And here we are going to look at the ENCODE dataset from the Snyder lab. Okay. So selecting a dataset takes you to its experiment page. This displays all of the information for that dataset. Across the top is the title of the dataset, what type of experiment it is. So here we can see that's an RNA-seq of mRNA baseline data, so unperturbed conditions. This also allows links to any publications. Each baseline dataset, or each dataset rather, is divided into separate tabs. In this instance, there are 4 tabs: results, experiment design, supplementary information, and downloads. When you go directly from the search page, the results shows you the expression for your gene of interest, in this case reg1b, within the particular experiment. Sorry, bear with us for a second. So, thanks. For this page, there are a number of features. Firstly, the bar across the top shows you the range of gene expression of genes within the transcriptome, in this case from 0 TPMs to over 1400. You can use the filters at the side of the page to specify cutoff levels. In this case, we consider anything below 0.5 TPM expression to be below cutoff. You can also change the data units from either TPM, transcripts per million, or FPKM, fragments per kilobase million. Looking at the heat map now in more detail, you can hover over points in the heat map to investigate further. For example, hovering over the point for Reg1b expression in the pancreas displays a mouse over with gene name, organism part, expression level, and number of biological replicates for that sample. Hovering over the data point in the heat map also highlights the position of the respective organism part in the anatomogram to the side of the page. Just to quickly note that there are a number of thumbnails by the anatomogram, and clicking on these changes the anatomogram to reflect specific genders or highlight particular tissues. The results page also allows you to search for other genes within the dataset. You can search for a number of genes by typing your gene of interest into the search bar and clicking apply. So the previous slides showed the expression of a specific gene within the ENCODE dataset we are using as our example. What would you need to do in order to see all of the genes within a dataset? So remember in the previous slide, in order to search for a specific gene, all you need to do is type it into the search box and click apply. The same is needed if you wish to see all the genes in the dataset. Simply clear the search results and then select apply once more. The heat map then shows the top 50 genes as rows, most specifically expressed across all tissues as columns. Sorry. Genes are ranked first by specificity, i.e. specifically expressed in an individual tissue, and then by expression level. This most specific view is highlighted in the green box, and this is the default view for a dataset. If you deselect the most specific tab, then the heat map shows the top 50 genes as rows, most highly expressed across all tissues studied. This is then ranked by genes with the most highest average expression across all tissues displayed first. You can also filter the data shown in the heat map on an experiment page further to display specific data of interest. Using the widget under the organism part filter, this creates a pop-up which allows you to specify in more detail the data you wish to display. For instance, you can filter on additional information captured in the experiment metadata. In this case, organism part, disease state, clinical information, sex, or individual. The next few slides are now basically going to highlight a number of helpful features we have for each dataset in Expression Atlas. The first feature is the ability to view your gene of interest in the context of a genome browser. To do this, simply click on your data point of interest, which then gets highlighted yellow, then click on the Select Genome Browser to view tracks button. Selecting from the drop-down, you can select the genome browser of your choice from the list. By clicking on the Ensemble Gene Browser, this redirects you and you can visualize the genome tracks, including alternative splice transcripts. Another great feature is the option to find similarly expressed genes to your gene of interest. Simply enter a gene in the search bar and click Apply. At the bottom of the resulting heat map, you will find a button called Add Similarly Expressed Genes, which you can click. When you do this, the resulting heat map will display a list of genes with a similar expression pattern to your gene of interest. You can use the slider bar at the bottom of the heat map to see more or fewer genes. Okay, so unfortunately, the next feature which I'm highlighting is not fully available yet for human datasets. This is the Show Box Plot and Transcripts view, but this is hopefully coming soon. So this dataset is a pig baseline dataset, similar to the human baseline dataset we've been using as an example. In this instance, I've searched for the gene KRT8, and I can see it is expressed in various tissues in the pig. Above the range of gene expression bar, there is a button called Show Box Plot and Transcripts View. Clicking on this, you will see two graphs. The top graph is the variability of gene expression across all biological replicates, analyzed for each tissue to give an idea of the variability between replicates. The second bar graph, which is only available for non-human datasets for now, provides an overview of the expression of individual known transcripts. You can either view them together or click on a specific transcript ID to select only those that particularly interest you. I'll now hand over to my colleague Anya, who will guide you through differential datasets.
Okay, so let's have a look at the second part of Expression Atlas that lets you explore differential expression. So for example, you can find answers to the question, in which conditions are genes differentially expressed? We'll start again with the left-hand search box and type in a gene of interest. As you can see here, you can either enter one or many genes, and you can use gene names like I've done here, or you can also use identifiers. And this will return results that match to any of those genes. That means it is an OR type of search. After you press search, you get to the differential results by clicking on the differential expression tab. This is a differential search results page. You see a list of search genes, and they are sorted by their logarithmic fold change value. And you can display this value by clicking on the button Display log2 fold change on top of the table. Each row in this table represents a comparison. This is a comparison of two experimental groups in a given experiment. So for example, here one comparison is called carcinoma versus normal. And when you hover over the comparison name with your mouse, you see a lot more information about the two sample groups that have been compared. For example, here you can see that the test group contained 10 samples, while the control group only had 4. You can see what was the genotype of the mice and which tissue was analyzed at all. In this case, it was skin. So if you want to narrow down the search results, if you haven't found exactly what you've been looking for yet, you can use the filters on the left-hand panel. Here you can specify a few things. So for example, you can choose to only show results from microRNA experiments, or only from RNA-seq experiments, or both. And the experimental variables filter lets you pick the type of comparison that was made. So for example, if you want to see if your gene is associated with a certain disease, just click the checkbox next to disease to only show experiments and comparisons where the disease state was investigated. So let's investigate this disease comparison a bit more closely by clicking on the comparison name. This gets you again to the experiment page with the title of the experiment at the top. The page controls here are quite similar to what we've seen already for the baseline experiment page, but there are a few changes. So for example, you can choose to only show the up or the down regulated genes. And you can also change the cutoff values for the log2 fold change or the adjusted p-value. And this lets you narrow down the list of genes that are being displayed again. So in this case, we only view the results for the 3 genes that we have initially searched for. And it shows us that the 2 of these genes are actually differentially expressed in this experiment, which was comparing the tumor versus the normal skin. If you like, you can download these results into a spreadsheet by clicking the download button at the top right hand of the plot. And here you have the option to only save the results that you're currently displaying on the page, or you can also download a full set of all the differentially expressed genes in that comparison. If you want to see all differentially expressed genes for that comparison, you can simply remove the pre-selected genes and press apply. You see this gives you the full list of all significantly differentially expressed genes. And then you can switch if you want to see only the upregulated or the downregulated genes. Okay, let's go back again to the start and explore another option. Instead of genes, you can also search for biological conditions. So if you don't have a specific gene that you're looking for, in the right-hand search box, you can specify a certain tissue or a cell type or a disease or any stimulation condition that you're interested in. This lets you find any experiment or comparisons that have been annotated with that term. So let's say, as an example, you are a plant biologist and you study rice and you're interested in the gene expression changes that occur in response to a drought treatment. So if you start typing the word drought, the condition search box suggests ontology terms. Just select one of the terms from the list to find all data points from samples that have been annotated with that term or any of the child terms. So this brings us again to the differential expression results page. This will show you all of the genes that have been significantly differentially expressed in one of the comparisons. Let's explore the first hit a bit further. In this experiment, we have 4 different comparisons, but the headers are a bit small to read, so let me magnify this for you. I hope you can read this now. In this experiment, there was a drought treatment that was applied to control. Drought treatment was compared and control treatment, and that was done in rice strains that are either susceptible to drought and strains that are resistant. And samples were taken after 3 and after 6 hours. So by default, you will see the genes in this list, and they're, again, the most specific ones. This means that the genes are only differentially expressed in exactly one of the contrasts. You can unselect the most specific checkbox to see the highest overall up or down regulated genes. This means these genes could be upregulated in all of the comparisons, like, for example, here. And they're not specific to any of the comparisons, which in this case means they are upregulated in both the susceptible and tolerant plants. So lastly, you also have the option to narrow down the view of the columns. So if you have more than one comparisons in one experiment, you can simply remove the other comparisons. And let's say in this case, you only want to see the 6-hour time points, so you can remove the 3-hour time points by unchecking the boxes and press apply. So the last thing I want to highlight about the differential results in Expression Atlas is the geo and pathway enrichment analysis. So if you go to the plots tab, you'll see for each of the comparisons several plots, and they show geo, interpro, and reactor enrichment analysis. And these are all based on the full set of differentially expressed genes in that comparison. So here's an example for the geo enrichment. And this shows you the top 10 highest enriched genes and how they relate as a network. So generally, for all experiment types, baseline or differential, you can also have a closer look at the experimental design if you go on the experimental designs tab. This shows you a table which gives more information about the samples. It shows you sample annotations and also has information about which experimental group each sample is in. And lastly, under the Downloads tab, you get an overview of all data that can be downloaded for a given experiment. For example, here we have the normalized expression data and also an R object that is ready for you to do your own data exploration in R.
Okay, I'll just finish by covering the getting help and sending feedback pages. So at the top of the Expression Atlas homepage is a series of tabs, and I'm just highlighting here the FAQ, the help, and the about tabs. Here you can find helpful information about how to use Expression Atlas and also how do we run the analyses for things such as differential conditions and gene set enrichment. You can also leave feedback via the atlas-feedback@ebi.ac.uk email address and follow us on Twitter @ExpressionAtlas. And I think that's everything, and we can cover some questions. I think there is a couple actually. Can we get the PowerPoint? Yes, the PowerPoint will be available. I think that will be up to Melissa, who will make that available shortly after the webinar is finished. With regards to the second question, sorry, just going to scroll back up. Can the log4.2 changes color scale be set symmetrically by the user? Unfortunately not. This is actually representative of the data as a whole. I think yes. So if the underexpression is much greater, say minus 10.6 to the overexpression, then the scale will be set based on the range of the data as opposed to by a range set by the user.
I think it's flexible. It changes as you display data.
Unless you have got one which is minus 3. Okay, one last question. Do you offer support or an API? Alfonso is going to like this. Or an AMI, images that can be deployed in AWS or host in-house proprietary data? I'm not entirely sure what AMI stands for, or AWS, sorry about that.
Amazon Web Services.
Ah, okay. So all of the data in Expression Atlas is generally public data. Some of the data sets, the raw data, can be found in Expression Atlas. The images are under a Creative Commons license, which allows them to be freely reused. For example, if you wanted to recreate the anatomogram, that will be freely available. You can also download the images and save them as SVG, PNG, or JPEG files. What are the other plant species that we can use this with? So if you go to the home page, just wondering if we can you will see a tab that says plants. And if you select on that tab, you will see all of the plant species that we host in Expression Atlas.
Yeah, we have quite a wide range of plant species, but I don't have the full list, so we can.
No, I think we have about 64. Not exact, I'm afraid, but something like maybe like 40 plant species, I think. It's been a while since I checked that actual number. So can we use an in-house installation of the EBI Expression Atlas? So the code to Expression Atlas is actually available on a GitHub repository. I think that's detailed in the help pages.
Yeah, and as we said, the data is also completely free and can be downloaded.
I think that is all the questions, unless there are any more. I think that's all the questions. Thank you very much for listening.
Just before we finish, just a quick reminder that this webinar is part of our regular Wednesday webinar series. There are a couple more webinars coming up in July, one on using to do with UniProt, the exact topic of which I can't remember, and the second one is on biostudies, which is related to Expression Atlas and ArrayExpress as well. Let's just do one last quick check to see if there are any last minute questions. Okay, so that seems like that's all for today. Thank you very much for joining us, and we hope to see you in another webinar again soon.
Thank you.
Thank you. Bye.